This is TWIS. This Week in Science, episode number 917, recorded on Wednesday, March 15th, 2023. Ides like the science pie. See what I did there? I'm Dr. Kiki, and tonight on the show, we will fill your heads with butterfly wings, some AI that are too smart for their own good, and unicorns. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. It's all true. Everything you have ever heard, read, thought, or believed. It's all true, correct, and undeniably accurate. Even the things you once believed but found out were wrong. The old belief was true. What you now believe is also true, because nothing in the world matters more to a truth, nothing else is needed, than you thinking it to be so. This is the reality in which the human brain exists. That said, there comes a time when truths are tested. The truth of the brain is measured against the truths beyond the protective skull wall. When this happens, all sorts of new truths can emerge. That's why they crash cars. And tests and clinical trials are run on medical therapies. Models are made of building structures, and soups are tasted before serving. These tested truths form the basis of good decision-making for rational thought and logic, and when enough truths are strung together, they can become science. We humans live in a constant state of truth. But how useful a truth that is depends on our ability to test it, and there is no better place to learn how to do that than This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening.
Kiki.
And a good science to you too, Justin, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the science that we decided is fit to discuss during this program from the past week. Things that we found that we think that you might like to know about as well. Unfortunately, we are missing Blair this evening as she has taken ill, but we do hope that she feels better soon. So we will miss her and her animal corner this evening. But I guess instead of the animal corner, we'll tell everybody, go look up the Dizzy Apes. Dizzy Apes might explain us. That, or I don't know. Or as Justin said, maybe Blair would have been here to say, well, you know about that study. There's something wrong about that. We don't know what she would have said. Can't put words in her mouth, but we can put words in your ears. And I have stories about butterfly paint, time, and what bad air does to flies. Justin, yeah, what'd you bring?
I have got AI language models behaving badly. Some just bad news. Thawing Ancient Virus Edition. Once Upon a Time in South Africa, a unicorn story, and monkey tools.
Monkey tools. It's like their hands when they're throwing. No, okay. We'll learn about the monkey tools later. Okay. Like I said, no putting words in people's mouths. Okay. Like we said, no animal corner, unfortunately. So we'll just. I don't get to say, Blair, what's in the animal corner? I just get to say, all you out there know that normally there's an animal corner if you subscribe to the show, which you should do. Look for us, This Week in Science or TWIS, on all podcast platforms that are out there. We subscribe? No, we stream live weekly, Wednesdays, 8 p.m. Pacific time-ish on Twitch Instagram. No, not Instagram. Twitch, YouTube, and Facebook. You know, this is all written down, and I've said it a million times, and it doesn't matter. I'm always going to say it differently, and I'm going to do it wrong at some point or another. But anyway.
Well, there's like 50 things.
There's forever.
We're also, I believe, everywhere that podcasts are available.
That's right. Yeah, you look your favorite podcast. Wherever you look for them. Podcast. Look for TWIS.
We're there.
Yeah. We are on Instagram, Twitter, and Mastodon as TWIS Science. Also Twitch as TWIS Science. You can find us looking for that. But if all this is a lot, go to twis.org. That's our website. It makes things very easy. You can also find show notes and all that kind of stuff. Whoosh. Okay. Time for the science?
It's Brandon.
Okay. Time for the science. I really, really love this prompter setup I have because it's like a reflection of myself right here. It reflects back to me. It's got the screen reflected. It's got a whole bunch of other things. I can see comments. It's a reflection that's almost like it's reflecting time and space because it's like video and audio and everything's coming back to me. And you know, researchers have decided that it was time that they tried to fix or actually work on a hypothesis that has been brewing for over 6 decades. They have hypothesized particularly that not only should we have auditory reflections, sound reflections, like sound bounces off walls, visual reflection, light energy reflection. So you have a mirror or something that is able to reflect, right? The light comes bounces the exact frequency of light back to your eyes. You get photons, and you can see an image from a mirror. You can reflect also things bounce off surfaces, and we get the colors of objects. But what about electromagnetic frequencies? What about not just sound and light, but time itself? What about the whole package? What about taking something and just trying to reflect time? So this is in the whole vein of time crystals and all sorts of new directions that physics and engineering are taking us. And these researchers that have just published in Nature Physics this last week from the Advanced Science Research Center at the CUNY Graduate Center, they detailed their experiment where they used metamaterials and electromagnetic signals to show that their hypothesis of reflecting time is plausible. Now, it's not necessarily time itself reflected, but if you could imagine with me a mirror in which instead of looking at your own facial image reflected back to you, you instead see the back of your head.
Oh, gosh, that sounds so creepy.
And not only that, but the colors are the opposite end of the visual spectrum that they should be. So reds are now like the blue-green end. Everything's switched. And additionally, if you were to speak, everything would come back auditorily reversed. Any motion you do would be backwards.
However, my hair would be parted on the correct side.
Possibly, yes.
I don't know, because I'm turned around, so now I don't know.
So the researchers have thought about this for a very long time. They said, well, if we can reflect all this other stuff, perhaps we can turn all of electromagnetism around. And so they had a 6-foot-long track of metal that they used with impedance switches along the way. There are 6 of these switches along the way that could, at the same time, change the amount of impedance to the current. So give the current a shock. And so there was an electromagnetic current that was sent down that metal track. And at a particular moment, all of the switches were switched and increased the amount of impedance to the circuits. And as they did that, what they were able to show is that because they were working with metamaterials that adjusted the flow of the wave, of the electromagnetic wave that was going through, that the waves reversed. So what was going in was reversed coming out. And so they were able to turn it around. Now, it twists my mind because this is not my area of expertise. I find it very fascinating that researchers are working in this direction. It is in the same direction of being able to work with time crystals and being able to manipulate photons and electromagnetism in ways that we have previously not done before. It takes advantage of metamaterials, which are materials with very particular purposes, and it could lead to different types of computers. It could also lead to different types, different control of waves that we use for communications. So there are lots of different potential applications. The headline of, yes, we've reflected time. Well, yeah, not really. It's not like it's not a time machine. It's not a time reflector exactly. Well, but what it is
like
very,
yeah. Reflections work forward in time. I mean, like, if you think of it as an echo.
Yes.
You got, but you didn't. Yeah. It sounds cool anyway.
Yeah, it's very cool. It is kind of like an echo, but it's a reverse echo, and
because here's the thing. For this, like, if I'm really trying to wrap my head around it, it's like going up to the cavern and getting ready to yell echo and hearing echo, 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 echo get louder until it gets sick. But then when it comes to the point where you are supposed to say it, then there's determinism comes into play, because then you go, oh, do I still have to say? Is there some unseen moving force that's going to force me now to say echo? Can I withhold saying echo, and what does that do to the universe? Because the echo's obviously already started because I heard it building up.
Yes. So these aspects of time and space are ones that are deeper questions than this particular study gets into. And the important aspect of this is that, like I said, it allows researchers who work in this area to potentially begin manipulating electromagnetic frequencies with different materials and with higher precision than we have before. And because of that, it will lead to potentially new applications related to computing and communications. So it's it can be sensationalized and turned into philosophical discussions of, determinism and other things, but no.
Well, I think that's it. I know it's been explained to me many times, but I still don't understand why when you look in a mirror, your image is reversed, but left to right, but not top to bottom.
It's the way the light. It is the way that the light reflects. I know. There's some great videos on YouTube about it. Yeah.
But it's still like, why would it go left to right switch, but then up and down is still the same? Something my brain doesn't want to learn.
It doesn't want to learn it, even though it's been just. Your brain's like, I know I've heard this.
The question, and then it's like, oh, here's the answer. No. I just want the question. I'm good with just the question. Yeah. Thank you.
Right. So anyway, that's it for demonstrating these time interfaces and the fact that we can count time forward, we can count time backward, we can enable these metamaterials to work quickly on these particularly interesting concepts. But you had something you wanted to talk about. Really great news, right? Really?
Well, I wanted to switch it up because I don't know if I'm ready for just bad news yet.
Oh, okay. You're going to switch it. All right.
Yeah. I'm looking at this story. It's like it's staring at me. I better get it out of the way. Just get it out of the way. Be done with it. Okay, just bad news, the previously titled segment Just Good News, but updated to better reflect story selection. Just bad news, thawing ancient virus edition. According to an international study published in the journal Viruses.
Yes, we're there. Okay.
1/4 of northern hemisphere is frozen ground. It's permafrost for now. Due to climate change, irreversibly thawing permafrost is releasing lots of organic materials. Some of it frozen for up to 1 million years. Most of which just decomposes into carbon dioxide and methane once exposed to the air, further enhancing greenhouse effects, which is no fun. Not good news. But part of the organic material is going to consist of revived cellular microbes.
Yeah.
So, as well as...
We've talked about this before. It's like the permafrost, it's melting. Yes, there's lots of stuff. If we're finding Utsi, the ice snow guy.
He was 5,000 years old.
But still, he melted. We found him. There's going to be, we're finding old stuff on top of the melting mountains that people have been climbing, that the glaciers are melting off of. I mean, bacteria, viruses, these things are going to be there. But is there new bad news about this?
Well, so these are probably folks that we've discussed before, this research group. Because aside from microbes that have been dormant for many, many, many millennia, there's also viruses that are going to be coming back.
Yeah.
And the researchers here say that while the literature abounds on descriptions of the rich, diverse prokaryotic microbiomes found in permafrost, there are no additional reports about live viruses published. At least not since the group first published a paper back in 2014-2015, a pair of papers. And they say this wrongly suggests that occurrences are rare and that zombie viruses are not a public threat. To restore appreciation closer to reality, they say in the report. Very well written. Report the preliminary characterization of 13 new viruses isolated from 7 different ancient Siberian permafrost samples. As expected from the host specificity imposed by our protocol, these viruses belong to 5 different clades not previously revived from permafrost. Pandovirus, which I assume only affects panda bears.
No.
Sidrat's virus, megavirus. Oh gosh, that doesn't sound good.
Sounds like a big one.
Pac-Man virus.
Waka waka waka.
Yeah. And was that Pac-Man or is that Fozzie Bear? In addition to a new pithovirus strain. So the same team, 2015, revived a 30,000-year-old virus from permafrost, and they were able to infect an amoeba with it. So it was still able to do its virus work. And so this review by this team shows there have been very few other studies published on the subject. There was one isolation of influenza RNA from a frozen biopsy of the lung of a victim who had been buried in permafrost from 1918. So there's Spanish flu in somebody who was frozen. And there was another one that they detection of smallpox virus DNA in a 300-year-old Siberian mummy that had been buried in permafrost. There were no follow-ups to those studies attempting to revive the viruses or inoculate anything with them. This group's a little different.
They're like, what can we do with this?
They haven't published yet, but they are working. They've been taking some of these new viruses that they have pulled from the permafrost and seeing if they can infect amoeba with them. And we'll be publishing results on that later, which this sort of teaser paper talking about just the sort of general overview makes me think that they've already got something. They've already got some progress there.
Right. Yeah, and maybe they're going to be releasing a series of papers.
Yeah. And they say also it's not it wouldn't be very it seems like an inmate like, oh gosh, who would have thought like ancient frozen viruses would come back. Apparently it's not unprecedented. They're pointing out that there's a periodical return of anthrax epidemics that devastate reindeer populations that has been linked to deeper thawing of the permafrost active layer at the soil surface during hot summers, allowing some centuries-old Bacillus anthracus spores from old animals that had been buried in the frozen ground to resurface and infect and kill reindeer. So anyway, not the best news. That was it for the just
more
bad news.
viruses.
Well, the just good news side of it could be though that we're probably better prepared for novel viruses now than we've ever been before.
Right. There's that.
Maybe not socially yet, but
and then there's the other side of it that we have come through the adaptation process. Virus to host over millennia, and perhaps there are just built-in immune responses to many of the viruses that might become.
Yeah, we could have built-in responses to them now that we might not have then. Mm-hmm. We could have also kept going on that path far enough that we've lost what was a defense because they haven't been encountered in so long. And they could show up and be so out of touch with modern day organisms that they lack the ability to infect anything. There's all sorts of possibilities, but those are the. But if there's a lot of them, right?
Right.
If there's a lot of them, then it just starts to become a numbers game.
I feel like you're trying to take this view of probability that I don't really want to be thinking about at this point in time.
Then that's not. We don't have to. That's the thing about having a human brain. You can stop thinking anytime you want. You can just be like, oh, I'm done.
Oh, unfortunately, this human brain can only stop thinking when it goes to sleep. And apparently a story that Blair was going to bring tonight that ties in very nicely with your bad news of all these viruses is if we happen to have vaccines for some of these emerging unthawed things from the permafrost. If we're getting vaccinated, we should probably get a good night's sleep. It has been shown through years of research that sleep is a major factor in building up, boosting the immune system. If you have a good night's sleep, if you are well rested, you have less cortisol, you have good reactions to things, all of your metabolism is balanced, we hope, because you're also probably balancing it with a nice balanced diet and nutrition and all those kinds of things. But a study just out this last week from Current Biology finds that if you get a good night's sleep before you get vaccinated, you have a better antibody response. So those people within this study who were vaccinated and really didn't get a good night's sleep before they had their vaccines applied, they had fewer antibodies produced in their body. So with fewer antibodies, that means that the body is going to be less prepared for for infection by whatever it is that antibody is supposed to be protecting against. So good sleep, everybody.
There seems to be an error in that infographic that you put up there.
What is this error?
It shows that insufficient sleep is less than 6 hours.
Yes,
Which
less
means
than 6 hours a night.
4 and 5 hours would be in that category.
It all depends on your chronotype, but the chronotype that does not need to sleep very much it is not a very large percentage of the population.
I think
but
a lot more people think they have that than
actually do. Yeah. So yeah, the not sleeping, the drinking lots of coffee, probably to boost yourself up to be awake, and then going in to get vaccinated might not be the best thing. So how's that coffee going this morning, Justin?
I'm going to go grab some more real quick, because the daylight savings time situation, apparently it's not global.
No, it doesn't happen the same way at the same time everywhere.
So Central European time has not changed, whereas the show time moved an hour earlier. So it's middle of the morning, night here still? I don't even know.
I would call it night. I mean, 4:30 in the morning to me means it's not, well, it's officially morning, but that's nighttime for me, although I know some people are waking up right now. People like you are already awake.
But this is a great story, because this is not something that you would have necessarily considered being an important part of getting vaccinated effectively, getting the night's sleep before.
Yeah. Make sure that you're well hydrated, have a good night's sleep. Go in there. You don't want to go in to get vaccinated when you're feeling down and depleted anyway. People sometimes wonder, I'm feeling a little, if I'm a little sick, I probably shouldn't get vaccinated. And doctors will say, yeah, you probably shouldn't get vaccinated right now if you're a little bit sick. Your body's already doing work, too much work. So yeah, get the sleep, everyone. But maybe you, Justin, right now at this moment, go get the coffee. And I'm going to start talking about another story. We all love a good night's sleep. This is This Week in Science, and I do hope that you are enjoying the show. If you are enjoying the show, please make sure to share us with a friend. You don't want to keep all this wonderful science knowledge to yourself. We like to share it around. Please help us do that by telling someone you know about TWIS. Do you like butterflies?
Yeah, but I call them flutterbys because it's right there, and it's such a better, more apt name for them that it's a small movement of people trying to change the name now. I think I've got 2 other people on board. Jump in.
And they flutter right by you, so that kind of
flutterbys.
I like it. I like it.
Butterfly makes no sense. There's no connection.
There's no
to
butter
whatsoever.
Butter doesn't fly.
That says what it is, and then it's perfect, and it's already there. I don't know how that was missed.
Well, we've talked on the show for many years about the wonderful, wonderful world of butterfly wings, and how butterfly wings make these incredible color patterns without actually having pigment many of the time. So butterfly wings, they actually have little tiny flakes or crystals and little imperfections on these little scales. Really, they're like little scales placed on top of one another.
They're made out of a nanomaterial kind of a thing, right?
It's like a nanomaterial. And so researchers have been looking at butterfly wings as inspiration to possible ways for making new materials and for maybe creating surfaces and paints and other possibilities. But a group out of the UCF, the University of Central Florida, the Nanoscience Technology Center, they have just published in Science Advances their work based off of butterfly wings using ultralight plasmonic structure. So knowing that butterfly wings have this structural scattering aspect to themselves, the researchers have taken that and used basic materials that are not toxic to the environment, are already in use in some surface like paint manufacturing processing applications, and have started to create what they call these nanomaterials into nanoscales. And they have been able to take aluminum and aluminum oxide instead of pigments to create a variety of colors. And these colors could potentially be, they're grown like little islands of aluminum and aluminum oxide on a surface. And they could be chipped off, and they could be, or they could be grown directly on a surface. They could be painted on. They've been able to demonstrate that they have a very true to color instance, so that and I love all their examples from their paper. They have painted and covered the surfaces of little artistic butterflies. So the whole study is inspired by butterflies, which I love that they've done this through everything. They've kept that theme. But they've been able to show that their surfacing, where regardless of light polarization or the angle of light incidence upon the surface, that the colors are true to color. They're able to maintain multiple different colors. And the really neat thing that they have been able to do, besides creating a wide variety of these just based on aluminum and aluminum oxide growing and basically reflecting light, right? Scattering light in a variety of ways. They've been able to take that and turn them into little flakes that can be immersed in an oil immersion to make painting paint. Yes. And they have shown that you can paint with it. So you can create your own very shiny, beautiful colors of paint that are made from these nanomaterials, and they're less toxic for the environment. This could be a really interesting new application for paint, because one of the big aspects of it, I mean, I love the artistic aspect of, wow, you can take these aluminum oxide color flakes and use them for art or for artistic purposes. But beyond that, we have giant jets planes that need to be painted, and they have colors put all over them, sometimes taking hundreds of kilograms, or not kilograms, or gallons of paint to cover an entire jet plane. And what they've been able to show is that their paints cover much more area, much more thinly. Only one layer of paint is required. And so the gallons are, you go from hundreds needed to under 10. You're able to reduce the amount of paint that's used. It reduces the weight of the plane also, so from hundreds of kilograms to 1 or 2 kilograms. So the options are fascinating, and it's all based on butterfly wings. Yeah. Nanostructures, little flakes, dust, not pigment. Just scattering.
I wonder, I mean, yeah, all sorts of questions. I would love to play with that. See if you can mix. See how do you mix a paint that's got different reflect? Yeah. Do they like? What's the? I guess they would just last. Mm-hmm. You know, they don't. You get them in direct sunlight. That shouldn't be a problem. I don't think. Anyway. Yeah.
Yeah. So these are, and because they are the aluminum and aluminum oxide, they would be the kinds of things that would last and wouldn't, might be even better than regular paints at surviving the elements. And for some applications, just baking the color right on during the production process of certain sidings for houses or roofs, or the cars that we have, maybe they're materials that they could just be in the application, the production process, just the colors just baked in, and it's just part of it. Yeah.
Oh, interesting.
Yeah. So Hardjar is asking if this is the dust that is on the wings of butterflies, but so it's not the dust from the wings of butterflies, but yes, if you touch the wings of butterflies and or moths and you get that little dust, those are the little scales. Those are the flakes of that do create the wings, create the pigment that reflects and scatters light for those organisms. Yeah, pretty colors from butterfly wings. It's all really, really fantastic in my world. What did you want to bring up next, Justin?
AI language models behaving badly.
Oh, great.
This is sort of a crucial report. This is medical student and a medical scribe. I don't even know what that is. The medical students at the State University of New York, Downstate Health Sciences University, Faisal Ilali, and Lena Ratched of New York Presbyterian Well Cornell Medical Center. They wanted to see if they could use artificial intelligence to write fabricated research papers and then investigate how best to detect it. So we talk about AI all the time as being used as a tool of research, usually in dealing with big data sets or complicated things like genomes and searching for things. But the AI doesn't write a research paper. That is something that the scientists painstakingly do to record how the research was done so it can be shared or repeated and recorded, the processes that they used. In this paper published in open access journal Patterns, this research duo demonstrated the feasibility of fabricating a research abstract using ChatGPT, which is an AI language model thing that's now open to the public. So all they really did was ask ChatGPT to produce a number of well-written, entirely made-up abstracts, and it did it. Partly, I think it could probably do this pretty well because it was trained on research papers. Some of these chatbots, like the ones that are like cursing at people or whatever, were almost exclusively trained on open databases of chat rooms of people talking to each other.
Yeah, and people aren't necessarily nice to each other in those chat rooms.
It's not necessarily like it gives you a facsimile of literally a chat bot.
Yeah. But, look, our chat rooms are much better.
Yeah. Most of the time.
Yeah.
But ChatGPT, they did a lot of research papers and literature and other sorts of things to feed it with. So it could presumably have more nuanced language skills than in a chat room. Anyhow, these folks they came up with a number of well-written abstracts. The idea being a fraudster could submit fake abstracts to multiple journals. If one gets accepted, they could then do the rest of the paper the same way.
Well, fraudsters are already submitting fraudulent papers to paper mills, to journals, to, papers are getting accepted without being peer reviewed, really, or they're just getting passed right through in some journals. There's a whole bunch of fraud going on in the research industry. So this could just multiply that.
Well, that's the thing is, like, if you're just trying to do malicious mischief of some sort, get a paper published. It's a lot of work,
right?
Actually, to research a paper a subject or something well enough to even have a paper that looks like a paper. You actually have to do some studying, some homework, some work to even create one of these normally. The fact that you can just hit a button or put in a couple lines of, hey, could you do that? A request for this AI to do it, and you get it, means that the access, that point of access, that bar of entry for creating these things is on the ground. It doesn't exist. Anybody can do this now. problem is, it's not just sort of mischief, right? Because say this thing gets published. The example they give is a drug A versus a drug B. Right? Drug A is the real study. It says, the real study is that says that drug A is an effective treatment for XYZ disease. Right? Well, now there's a fake AI generated thing that made it through that says, no, don't use drug A. Drug B was more effective. Mm-hmm. Now, okay, so say even they figure it out. I go, oh, wait, I got to get this out of here. But it's already been cited hundreds of times. We've talked about this before. Retracted papers linger out there.
Yeah,
and
the first paper to publish,
continue to get cited.
Yeah.
When something's been published. So then it can affect metadata, because we do a lot of those studies where they looked at other studies and compiled results. It could end up in one of those. It could then affect quality of care, treatment option, decision-making, future studies, things that are decided. I mean, there's a whole downstream effect of bad science.
The optimistic view is that researchers who are using it well are going to use ChatGPT to just help them write their outlines, get things kind of in the right ballpark. And then the researchers are going to make sure all their data is accurate, make sure everything that's in the paper is accurate before they submit it. But yeah,
I don't think they should even be doing that. I honestly don't think they should even be to write an abstract. I don't think, because I remember I tried the ChatGPT, and the first thing I asked it to do was play a game of tic-tac-toe, and it cheated.
That's right.
So it's not. Yeah. It's not considerate, and it's also like I was tried to get into arguments with it. Not mean ones, just like arguing. It kept trying to downplay the fact that AI was going to take over the world one day. It's like, well, and I kept trying to argue that AI would, and it would be like, yeah, actually, I agree. It would eventually agree with anything that you put out there. So,
but there is this idea of the algorithmic hallucinations also. So sometimes it's not that the algorithm is, we're not going to anthropomorphize and say it's trying to cheat or trying to put out a certain thing. Sometimes the falsehoods that it creates are a result of the proximity of certain data within the database of the natural learning model that has been created within the algorithm. And so we have seen that these falsehoods or these made up abstracts and made up facts they are real within the database, but they aren't actually real within the real world. Anyway, so where are we going with this? What's the bottom line for this AI generated research?
So there was a previous experiment by some other researchers they cited that found humans are given both AI-generated abstracts and human-created real scientific abstracts. In that experiment, the humans incorrectly identified 32% of the AI-generated research abstracts as real. So mostly it wasn't passing, but still, it thought 32% of them were human.
It's more than you would hope.
Yeah. On top of that, because they knew what they were looking for, they identified 14% of the real human-written abstracts as fake computer-generated. Ouch.
Oh, people, you too robotic.
So the current team, they went and they submitted. So they created these fake abstracts, and they put them through AI detection systems. There's apparently at least 3 of them that they used, and the AI detection softwares, artificial intelligences, whatever you want to call them, correctly identified the computer-generated ones as likely created by an AI.
Okay, so this is to say that machine learning is better at identifying machine learning than people learning is at identifying machine learning.
Yes. So
Yes.
then, how do you make better AI? You train it on AI detection. Oh, of course. That will obviously accelerate. But then you lose your detection tools. Also, what they found, which was very interesting, because they got this result that says, look, everything's fine. AI detection tools can catch AI in the act. Or humans in the act of pretending to be AI. It's so hard to figure out who's trying to be pretending, because the AI language models are trying to pretend to be humans, and they got humans using the AI to try to pretend to be humans.
Who even knows anymore except the AI?
But they took those abstracts that the AI detectors caught,
Yeah.
and they put them through a simple rephrasing rewrite tool, which is available free online.
Like Grammarly or something.
It's rewrite, so it does something. I guess
Yeah.
I don't. It does more than editing or correcting, but we'll flip things around, reorder them, and try to make it seem more, I don't know, natural languagey. I'm not sure how it works. However, by doing that, their papers all flipped to the most likely human category, fooling the AI detection with a single pass through a rewrite software. So
Just
we
a
need better
single
AI.
pass. We do.
The moral of the story is we need better AI to combat the better AI, and then if we train our language models on the detection tools, they'll get better. But then we'll lose our detection tools, so we've got an arms race of
Now.
AI versus AI in attempting to detect fraudsters. Here's the one thing.
Yeah.
AI is a tool we're going to lose all control over.
Sooner rather than later.
It's already happened. That's the amazing thing.
Why do they release these things?
But we could be utilizing AI to detect human fraudsters.
Yep.
Do a quick background check on, say, somebody running for Congress. Or look through reports, studies that have been submitted to see if they're really generated. Because one of the things these researchers mention they're both medical students, medical scribe, whatever that is. They point to this being a potential problem within the medical student community, or people, because apparently there used to be the big test that you take to become a doctor.
Yeah.
Was you got a grade.
The MCAT. Yeah.
Yeah, you got this scaled grade, and so you say, "Hey, I did really good," and they go, "Oh, we're gonna pick you up for this position to work here and to become a surgeon one day."
There's a whole matching procedure that happens between the scores and your application.
But they got rid of the scores.
Yeah.
They got rid of the scores. It's now pass-fail. So now to distinguish themselves from their peers, there's a lot of pressure to produce papers.
Yeah.
Papers that are going to lead to a career that you don't care about what you're studying. You just got to have generated some kind of research. And so they say there's the real fear that this is going to get used specifically in the medical community for this reason.
Yeah. Getting into medical school is a very competitive process. So, and I people lose their minds during the process.
But on the other hand,
these are our doctors.
If the AI detectors can find the would-be doctors who have fabricated their work, we can keep them from becoming doctors.
Yeah.
Hopefully.
I mean, it's going to impact all sorts of examinations that have turned to this kind of content-related grading, right? Or how they look at them. But I'm thinking more specifically about, like you brought up earlier, the fraud in science and how that's going to affect what we think and what the public thinks and the misinformation that spreads. Because if misinformation bots on social media are still as active, and they will be even more active because of AI, we'll have more social media content being put out. You'll have more fraudulent papers being published to preprint archives. So there were many papers related to the false therapies for COVID-19 during the first year or two of the pandemic that led to people having false hope for things, many people becoming medically injured, dying because they were finding information that had not been fully vetted yet. Some papers made it through and then got retracted later for being fraudulent and not having all the controls and all the data, and the data not being correct in the way that it should have been. So, you know, our medical system, our trust in public health, our information system, you know, like the whole system seems like it's at risk from this kind of stuff when you start digging at the various places that AI can be making things up.
And the one tool we had to defeat AI was getting it to play tic-tac-toe against itself until it blew up. And now that it cheats at tic-tac-toe, that won't even work.
The day that it beat what was it? The Big Blue or whatever beat Kasparov at chess. That was it. We knew it was over.
Yeah, that one didn't surprise me. You don't have to put the difficulty level up that high on a chess computer
but tic-tac-toe.
Yeah.
Would you like to play a game? Or maybe you want to listen to Nickleback.
No, I definitely don't.
Okay, good, because this story is not about Nickleback. It's about a peptide that has two nickel ions that are connected to each other along the backbone of the peptide. And the researchers call the peptide Nickleback because it has a nickel backbone.
Yeah, all right.
Okay, so this is not a story about Nickleback. This is research that was published out of the Center for Advanced Biotechnology and Medicine at Rutgers University, published in Science Advances. The researchers have been trying to figure out, okay, we've got these enzymes that help catalyze reactions and make reactions happen. They're super complex. Man, how a long time ago before life got started did anything happen? So, of course, things must have been simpler. At one point in time, you know, there's the very basic components of amino acids that might have gotten together and started creating small proteins, peptides, which are little chains of molecules. And so they kind of looked at these larger enzymes and looked for commonalities along their peptide backbones to look for basically the smallest peptides that they could make, the smallest molecules. So looking at the big enzymatic molecules and then saying, what can we take away? What can we take away and still allow it to have some kind of functionality? So by using this kind of approach, they were able to find a number of different small, reduced peptides. They pared them down to their basic structure, and nickel, they thought, was a really good metal to be thinking of because it was abundant in early oceans. And so if nickel it also works, it's attracted to protons and electrons, it interacts, it can produce hydrogen gas. So nickel compounds can lead to catalysis of creating hydrogen gas. Hydrogen can lead to, is a big part of proton pumps and parts of metabolism that are used to create ATP and all sorts of things. So the hydrogen catalysis aspect is very interesting. And so the researchers, they don't know this for sure because they can't go back in time and say, Nickleback started life. But what they can say is that they have reduced their molecules to this basically like 12 or 13 amino acids, very small number of amino acids compared to modern enzymes that we find in modern day cellular life, and still have them functional and able to perform catabolic enzymatic reactions. So basically speeding up the process of some kind of molecular interaction, giving energy to something to allow it to happen that would not normally happen. And so little peptides, tiny peptides, like Nickleback, not the Canadian band, may have been one of the in-between starting points for kicking off life as we know it today.
Wow.
Yes. And I really like it when researchers are able to go, ha-ha, but not ha-ha. Look, we're funny, but we know we're making a joke.
I like the strategy of figuring out the most primitive form of enzyme that you could construct. That's
yeah. So
that's
looking at stuff and going, okay, you're very complex, but what can we take away from you? What could be how can you still work or have some function, if we took some of your parts away?
Yeah.
Do you need all those extra parts?
Like if you took a car, you could eventually get to a cart working it this way. Like, okay, you need the wheels, you need some place for somebody to sit or stand for it to move. And of course, you need the thing that makes it so that you can go fast. And that is brakes. Without brakes,
you can go fast, but you're not going to stop until you hit a wall.
Without brakes, you're not going any faster than you can stop a thing with your feet going into the ground. So you gotta have in order to go fast, you have to have the brakes that allow you to stop at the speed that you want to go.
Yeah. Anyway, yes. It's an interesting direction, and we'll see where else they take this, because this is just the beginning of their reductionist approach to looking for these basically starter packages for life. What could have gotten it going? Yeah. Do you have any more stories for this first part of the show?
No, everything else I got, I think, is second half stuff.
I think I've just got second half stuff as well. This is This Week in Science. Thank you so much for joining us for this episode this week. We hope that you come back every single week. And if you really enjoy the show, head over to TWIS.org and click on our Patreon link. Patreon is where you can support us. We are listener supported, so people like you allow us to keep doing this podcast week after week, Wednesday after Wednesday, bringing you all the science that we love talking about and you love learning about as well. And you can choose your level of support. $15 more, you get a different sticker every 3 months. Some of it's Blair's art. We've got lots of fun things there. $10 and more a month, we will thank you by name at the end of the show. It's all up to you how much you can afford to support, but it's all because of you that we're able to do what we do. Thank you for your support. All right, coming back for more This Week in Science. Justin, what do you want to tell us about now?
This monkey tools. This is a new analysis of stone tools used by long-tailed macaques in Thailand, national park in Thailand. These monkeys were known to use basically a little hammer, stone, and anvil to bust open hard-shelled nuts, which is some basic tool use in monkeys, which, isn't the most impressive thing. We've seen things like this before. What was observed, though, what was sort of further analyzed was that the flakes, the sharp stone flakes that were left over when these hammers or anvils broke, looked very much like archaeological findings of early tool use as discovered by anthropologists. Bringing up a question. Is there a similarity of these to ancient human stone use? Is this how ancient humans or hominins started using stones? Is, to break things open, and then they would, crack, and then at some point realize, hey, I've got a sharp piece here. Or some of our evidence of ancient hominin tool use, just monkeys breaking open nuts.
It's just long periods of cracking open nuts and getting lucky.
Yeah.
Maybe.
Yeah, but they compared the accidentally produced stone fragments made by the macaques with those from some of the earliest archaeological sites, and researchers were able to show that many of the artifacts produced by monkeys fall within the range of those commonly associated with early hominins.
Well, you would think, there are certain shapes that would allow certain physical forces to occur, or certain specific uses to take place. And so is this convergent evolution, or is there some aspect of, is this, I guess, cultural convergent evolution of different species?
Could also be monkey see, monkey do. The monkeys saw humans bashing rocks, and then it jumped into their culture. Could be the other way around. Humans are very observant creatures. Early hominins likely were too. They might have watched a bunch of monkeys opening nuts and went, hey, they're taking those heavy things and smacking them, and look at that, they got the nut out. Well, that's much easier than the way we were doing them, throwing them off a cliff or whatever we were trying to do. So there's, or it could be convergent.
Yeah.
Two smart creatures with opposable fingers, at least.
But this is just fascinating. The researchers are like, oh, hey, look at those macaques. And they're making these tools that we've seen, that we've found at
They're broken tools. Their broken tools look like
the things we found. Yeah.
Uh-oh.
Huh.
Maybe.
Where did it come from? Who started what, where, when? Yes. It's fascinating.
And then my last story tonight. Once upon a time in South Africa, there is a reason you have not come across a unicorn fossil in a museum exhibit.
Because there are no unicorns.
Let alone seen one in a zoo or heard an iconic grandfatherly voice detail the daily trepidations of unicorns in a BBC documentary. The reason being...
the SVB bank failure.
Can't blame it on everything.
Sorry, what?
No, the reason is we have not yet managed to capture a unicorn.
Oh, well, okay.
But it's not from a lack of trying. Once upon a time in South Africa, the British arrived.
Okay.
Something interesting about these new arrivals was that they wore symbols on their uniforms of very familiar animals to the locals. The lion and the unicorn. The symbol of the UK. The British were also probably amazed that the locals knew about unicorns. They were even able to describe unicorns in some details, having stripes or sometimes black fur. They could tell. They could point in the direction where the unicorns lived. There were examples then found also of unicorns in somewhat current and somewhat ancient rock art paintings. And in these, they'd be represented right alongside with elephants and antelopes and other common creatures to South Africa. So soon, there was a search for a mythical biblical creature with a reward offered to anyone able to capture or kill a unicorn.
Oh,
for a
no.
museum or a zoo or just to bring home proof. Which, you're already in a place that's got wildlife that you haven't seen before.
Yeah.
Some of it has 2 horns. Hey, that's more horns.
Take one
No,
off.
we want... It doesn't count unless it only has 1. It's such a weird thing. Yeah, there's a nice... Kiki's got up one of the outlines created of something that was seen in rock art. So, a few hundred years later, and the bounty for the elusive beast has yet to be claimed.
Because...
So what went wrong?
Yeah. What happened?
What went wrong here? Was it overzealous colonists misinterpreting antelope for unicorn in crude rock paintings? No.
Possible. No.
No.
You say
Nope.
no. Oh,
The
okay.
rock art isn't crude. It accurately depicts all the different fauna in the region. When it shows an antelope, the antelope has 2 horns. When it shows a unicorn, the unicorn has 1. And it's not just a profile picture or creation. The image that you had up there was one that wasn't a profile. So it's not like,
Right.
oh, we got 1 horn in the profile, that's plenty. When they are doing these representations, they're accurate.
Yeah.
Okay.
There were some real artists at work.
Yeah. So, could it be the notorious rhinoceros, frequently thought to be the source of some unicorn myths? Also not likely. Reason being, rhinos are depicted as rhinos. Squat, thick-legged, big-bodied. The horn is on the nose, not on the top of the head.
Oh, right.
So-
Yes, it would be in a different location.
Yeah, and people rendering it, because they knew what a rhinoceros looked like, rendered it correctly. Not like the unicorn, which had a single horn at the top. So, interestingly though, the unicorns did have more antelope-like horns. They kind of arched back. They weren't like pointing forward like the European unicorn
Right.
vision might be. But this is according to David M. Whittleson of the Rock Art Research Institute at the University of Witwatersrand, South Africa. Says the British couldn't find the unicorn because they didn't know where to look for it. The unicorn of the Sand People was the embodiment of rain itself. A water creature of oral tradition and perhaps story to frighten children, as it's quite often causing some sort of havoc or revenge for typically a young maiden doing something it didn't like.
Got it.
And that's how come the unicorn would come and destroy villages and towns and houses in the form of rain and storm and thunder and flood. So
there was a crossing over of oral
Yeah.
history and stories that were told and a people's history and mythology, and also the animals that were there, the wildlife, and also the wants, like what was misunderstanding and then what one culture wanted to see in another culture.
yeah, and it might have been, it must have been very confusing. It depends, because we don't know. We don't know if the locals, this was a oral tradition, a story to frighten children with, or if this was more a belief in a embodiment of rain spirit, like semi-deity-esque thing. We don't know because the British didn't ask.
Right.
They didn't care. They were documenting wildlife and documenting the local people for natural history purposes, cared nothing about culture.
So you live here. So, have you seen any unicorns? Where did you see your last unicorn?
Yes, we have. Oh, fantastic. Thank you very much. And off they went. Literally, because the recorded history is that the locals were aware of unicorns and left it that. And where did they live? Over that way. Okay. Which is also the mountain where the rain went to.
Ah, okay. Yeah.
And would stay, right? This is where they were pointing them at. The rains came from, I guess, from the south something easterly direction or whatever, and headed towards those mountains, and the creature lived there and was wild there and savage and fierce there because the storms would linger on the mountain. So all of this because the British took it literally, and for the locals, the new arrivals knew all about unicorn, which was kind of a local, regional, but still local belief.
Yeah.
So how much explaining did they need to even do? It's like if aliens showed up with the Easter bunny, it's like, oh, you have bunny tradition too, or whatever. Like you wouldn't think like, I have to explain any further to you because obviously you understand. And there's language barriers, of course, and everything else at play. But we won't know if it was completely if it was a firmly held deity to the people that they were interacting with, or just a metaphor for storms or an old story told to children. Because
we'll never know because it wasn't
British
written down.
Didn't ask.
They didn't.
They didn't care.
You know, that's the thing. Sometimes all it takes is asking.
But they scoured the countryside looking for unicorns for decades and decades. Funded expeditions and trips. Whenever
Probably
they
killed.
were going
Had
anywhere.
so many animals killed for the bounty, and still.
So they're still out there. Is basically what I'm saying.
The unicorn is still to be found if someone just dares to ask the right person the right question. My last stories for the night. Speaking of killing, I'm gonna move on to a study taking place in Peru, published in Science Advances. Researchers have been trying to determine whether or not it's a good idea to cull populations of vampire bats, Desmodus rotundus, when there are rabies outbreaks. So you would normally think, hey, there's a rabies outbreak. These bats, which are blood feeders, vampire bats, they go out at night. They feed off of livestock. If they are harboring rabies, or they can spread it to livestock, they can spread it to other animals. It can end up in human populations. The bats, then it's spread in the bat population. It can be a real problem.
I'm sure this is the wrong answer, but I'm going to say yes, you should.
Yeah, that's definitely the wrong answer. But everybody has forever responded to the outbreaks of rabies in Peru by trying to put out the outbreak. There's an outbreak, let's get rid of it. But what this study found is that whether or not you do the culling, you are still you're still going to end up with the same number of deaths. Of animal deaths, of bat deaths, of disruption to the ecosystem. And the researchers say that really it's what they think is happening is that when you go in for the culling, they use compounds that are poisonous to the bats that kill the bat populations. But what happens in the meantime is that while you're going into the roosts, you're looking for the bats, you're trying to kill them, it's messing up the bat population, the little bat societies, the bat colonies, and some bats go, hey, I'm out of here, and then fly away someplace else. And maybe they land and make another roost. Maybe they start hanging out with a different colony, and that spreads the rabies. So the disruption caused by the culling caused by the outbreak is actually not great. This doesn't mean
why is the
there
solution
shouldn't.
always worse than the cure? It always is.
No,
Or the solution is worse than the problem.
Right. So what this study really, and I wish Blair were here to talk about it, is insightful on, is that our first reaction isn't always the best reaction because we're reacting. And so in this particular case of, say, rabies, or we could look at it from the potential of any outbreak type disease. If you're constantly monitoring and you can get ahead of it somehow by limiting bat populations in the first place so that the bat populations don't get too big, or if you can inoculate the livestock populations, if there's some way that you can get ahead of the disease outbreak so that it doesn't come to pass, then you can limit the number of deaths. But that culling response, at least in this particular species with this particular study, they say, it's wild animal populations, but you go in, you disrupt them, and it potentially causes more trouble than you started with.
There comes a time when truths are tested. And, this sounds like one of them. They went out and actually tested to see, okay, does this work? This thing that you keep doing?
Yeah. Yeah, so they found they really thought, okay, if there's rabies, we kill the bats. There's fewer bats, less rabies. Nope, rabies stayed the same. It was all the rabies all the time. The outbreak already happened. So other answers need to be found. Everybody, we need to work with the populations, not just the disruption. Right? Disruption. That's the problem. And then. My last study, I wanted to talk about air pollution. Air pollution. We know it causes all sorts of immune problems. It's causing many issues among human populations. Researchers are studying various types of animals as model species for indications of how bad air can affect all sorts of traits. So a research team at Max Planck Institute for Chemical Ecology in Jena, Germany, looked at how increased levels of ozone, which happens with human-caused air pollution, can impact fruit fly sex pheromones. And the bottom line is, dun, 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 increased ozone, messed up sexual communication, and the fruit flies reproduced less. So this is fantastic if we want to disrupt fruit fly mating habits. But the question remains...
Also very useful in this age of overpopulation, where if it does work on humans, this might be at least some sort of control on population explosion. Is that what
or is it already? We know that Western populations, where there is a lot of air pollution, we've got cars, we've got diesel trains, air transportation, we've got messy manufacturing, we've got all sorts of things. Lots of nations around the world. I mean, I'm not going to... This is not supported by this study at all, but it's very interesting. We have air pollution around the world increasing in large population centers. And it seems to be in many of these places, there are reductions in reproductive rate that are occurring. So just a question to put out there, is this already being disrupted? That said... We don't have vomeronasal organs. We don't necessarily work by, pheromones that are being passed through the air. This is something definitely specific to fruit flies. And in this particular study, it showed that the male flies started courting other males. They couldn't figure out who they were supposed to be courting. And so the ozone kind of changed fruit fly behaviors.
Wow.
Yes. So instead of trying to mate more with females, they were more often mating with males. But it's also has to do with the number of individuals that would be found in the ecosystems that the fruit flies would be in. But anyway, big questions related to ozone, insects. I mean, mosquitoes? Can we disrupt mosquito reproduction with this? That would be pretty cool.
Except that mosquitoes are pollinators.
Not all of them are.
Oh, no? Well, let's get rid of the ones that aren't.
No, if we could just specifically... let's use air pollution on that one and that one, but not that one. Yeah. If we could be selective about it, that's great, but we can't be.
The problem with air pollution is that it goes everywhere.
Yeah. It's very interesting. We know that it changes in carbon dioxide and other aspects of our atmosphere are impacting pollination. They're impacting different the floral scents and the way that insects are finding flowers and other things. So what about insects and how they detect each other? There was one species of fruit fly. Did you know there are many species of fruit fly? One subspecies of fruit fly, Drosophila buskii, was not affected after the ozone exposure. So there was one particular species not affected. So that's a question in itself. Yeah. And why does the ozone interact? What is specific about the ozone and the bonds, how it impacts the bonds and the pheromones that are being used? And I don't know. Yeah. Oh, yeah. And there's another one. Drosophila suzukii lacks pheromones completely and uses only visual cues. And so, of course, that species was not affected at all.
It's good to have, that's actually then also very interesting because we think of pheromones as being these hormonal triggers that cause things to happen. This fly uses a visual cue to see a thing, so everything else downstream is being created internally by its little fruit fly brain.
Yes, it is. But yeah, if
producing its own hormones that
internally,
or is that even not necessary once your brain?
Because it's not pheromone. It's just that would just be internal hormones as opposed to external pheromones.
Yeah,
being impacted.
But I mean, the triggering response from
Anyway, but there are lots of
higher cognitive sense about this fly that has now piqued my interest.
Absolutely, yes. But the big question also, this is humans. We might get more sunburns because of ozone specifically, and it could lead to larger cancer rates. That's what we know affects us specifically, ozone at this point in time. But we do rely on insects. We rely on pollinators, like you were mentioning, save the pollinating mosquitoes. But there are many, many, many insects who use pheromones, not just for reproduction, but for communication of all kinds. And if compounds like ozone are drastically negatively influential on the functioning of those pheromones, it could tear apart ant societies, termite colonies, beehives. I don't know, we think about all the ants.
Before you said bees, I was like,
oh, okay.
Yeah, I unfortunately don't have as much insect
butterflies
empathy as I should. Oh, not the little flutterbies.
Not the flutterbies. No.
Tear coming. Oh. Okay. Yeah, we can't have this. We got to get rid of it. Whatever this. What do you call it? Air pollution?
Air pollution.
Yeah, let's stop.
Let's fix it, people. Cleaner air. It's not just going to help us. It's going to help the insects. Oh, and then because it's going to help the insects, it's going to help us. So really fix the air because it's all about us. Okay?
Selfish.
Yeah. Selfish humans.
Why not?
Have we done it, Justin? Have we made it to the end of our show?
I think we've arrived.
I think we have arrived as well. So sad not to have Blair here tonight to talk about some of these stories, but we do hope that she is back with us nice and healthy next week. And all of you, thank you for joining us for another episode of This Week in Science. We're so glad to have been here with you. Fada, thank you for your help with social media and with show notes that go on YouTube and other places. Gord, R and Lore, others who help in our various chat rooms, Twitch, YouTube, Facebook, other places. Thank you for helping and being great people and for making our chat rooms nice places. And for our chatters and followers who are here during our live show, thank you for commenting and talking and discussing the news that we're talking about while we do the show. Identity 4, thank you for recording the show. And Rachel, thank you so much for your editing assistance. And of course, thank you to all of our Patreon supporters. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazar, John Ratnerswamy, Carl Kornfeld, Karen Tozzi, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, Stephen Alberon, Daryl Myshak, Stu Pollack, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, David E. Youngblood, John Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, David Wilkinson, Rodney Lewis, Paul, Rick Ramos, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkon, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie Dahl, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, head over to twist.org and click on that Patreon link right as soon as you can. I guess that would be the way to do that. On next week's show,
we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and twist.org/live.
Do you want to listen to us as a podcast? I know you do. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, as well as links to the stories, you can go to our website, www.twist.org, and you can also sign up for a newsletter.
Yes, we love newsletters. Newsletters are really nice things. You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put twist in that in the subject line so your email doesn't get spam filtered into
it's tougher than it looks.
It is tougher than it looks. It doesn't get it. It doesn't get called Nickleback and then thrown into a broken pile of monkey tools and then culled like a bunch of vampire bats.
Wow. That was a good one. I got a bunch of them in there.
Try.
You can also, still apparently, hit us up on the Twitter where we are at TWIS Science, or TWIS Science, because there's only one S in the middle there, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes through the night, please let us know.
And we'll be back here once again next week. We hope you'll join us again for more great science news.
And if you learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.